and good afternoon to many of you who are listening out there. And this is, another webinar in the series of webinars from Tribute to Life, the Commonwealth, the Tribute to Life project of Commonwealth. Today we have an excellent webinar on setting up immunology labs for transplantation. And we have two great speakers and we have two chairpersons. Our chairpersons are our International Advisory Panel members. We have Dr. Hasina, who is an intensivist from Trinidad and Tobago, and she is going to be a chair for the webinar, along with Dr. Kishore, who is also an International Advisory Panel member, and he's from Guyana. He is also a surgeon, he's a transplant surgeon, and they are going to be chairing this webinar today. So I hand it over to Dr. Kishore and Dr. Hasina to take this forward. Thank you.
Hi, good morning, everyone, and thanks for being on this webinar. It's my first time here, and it's my pleasure to be among you guys. We will all have introduced ourselves, and a brief statement about us. From Guyana, we would have started our program around 7 years now, and I'm happy to be part of this panel, sitting in for Dr. Margaret Touche. Dr. Hasina, you want to say anything on your side?
She has Dr. Hasina, I think we'll reconnect Kishore. I think you can go forward and introduce the first speaker if you want to.
So our first speaker is Dr. David Briggs, and he's from the UK, who's a consultant clinical scientist at the National Health System for blood and transfusion. So Dr. Briggs, and we will be presenting on the transplant laboratories in the Caribbean, how we can move forward. Thank you.
Thank you very much. I will see if I can share my screen. Okay, is that working?
Yes, it is working, sir. Just make it full
Wonderful. Screen. Okay, good. Thank you very much. Well, very kind of you to invite me to talk. I'm going to really give a talk about the why we need an HLA laboratory, transplant laboratory, what we do, and when we do what we do. The why, I hope, is familiar to everybody. The main reason is preformed antibodies. If you've got preformed antibodies, donor-specific antibodies against the donor, if you look on the left, this is what happens. You get hyperacute rejection, the kidney clots, and is lost within minutes. And there are two main types of antibody we need to worry about when we ask the question, does the recipient have antibodies against the donor? The first is ABO groups. And I wanted to mention these because of their contrast with HLA. The compatibility with ABO is predictable. If you're blood group O, you will have antibodies against A and B. As you can see on the right, green indicates whether that's whether you're compatible or not. O group patients can only accept O group donors. A patients will have antibodies against group B, so can only accept A donors, not Bs and ABs, but of course could accept O donors. And similarly for B group recipients, can't have A group donors. AB donors don't have any antibodies, so they're essentially universal recipients. Whereas O group donors can only have O group, so O group recipients, O group donors. All you need to do for this is ABO type donor recipient, probably confirm them, and then the compatibility can be predicted. HLA is a bit more complicated. The sensitization is not predictable. You need to be exposed to non-self tissue, whether that's a previous transplant, transfusion, or a pregnancy, to form HLA antibodies. The other problem is HLA does not, like ABO, just 4 different sort of forms. It's highly variable, and I'll describe that in a minute. This means that testing is complex. The antibody characterization is an HLA typing complex, and as is the cross-matching. So what do HLA look like? Well, there's actually two broad groups, class 1 and class 2. Each one have about 3 members. They're given letters. A, C, B, and C are the class 1. The DR series, DR, DQ, and DP are the class 2. These are all coded by genes closely linked together on the short arm of chromosome 6. They tend to be inherited as a block. And each one of these loci producing the individual proteins is highly variable in the population level. And we give each variant a number. So A1 is a different form than A2. And it's the combination of these and one set from each parent, which is your HLA type. So below here, you can see there's an HLA type given as A1, and A23, B7, B18, up to 2 of each, etc. That is a tissue type. It's far more polymorphic than that. And roughly to date, the total number of different variants across the whole of the HLA region is at least 25,000, probably more than that. It goes up every year. It's a little bit more complex than the previous slide, because the class 2 series tend to have 2 genes, an A and a B, which form an alpha and a beta chain for the protein. So there's an enormous number of combinations, and actually potentially there's billions of different HLA types combinations, although we tend not to see all possible forms. But if you look in a family, the variation is luckily much more limited. And this is just a sort of arbitrary pedigree, and I've just shown the A's, C's, B's, and the DRs. And in a setting where the parents have completely different HLA types, different combinations from each chromosome. But you can see on the left, the first two offspring have inherited the same combinations from each parent. So they're HLA identical. And about 1 in 4 siblings you'd expect to be HLA identical. But you can also have siblings which inherit completely different HLA sets from each parent. And again, about 1 in 4 siblings will be HLA non-identical. The remaining 1 in 2 should share at least one of the haplotypes. So how do we identify all of these? Well, when they were first characterized, we typed these serologically. We had a sera against some of the types, and we use a sort of microassay, which is read by eye and by microscope, to see whether you had a particular reaction. It was about a 3-hour test, so relatively quick. The problem is, this is very much limited by available anti-sera, and probably the resolution is no more than about 100 antigens. And we now know there's in the sort of tens of thousands. But as DNA methods became available in the early 80s, various methods were developed. There is sequencing, which tended DNA sequencing, which identified all the sort of the new forms. But there's 2 basic techniques which are reasonably rapid, about a 4-hour turnaround. One uses the polymerase chain reaction, so you need a separate reaction for each allele you want to identify. But even so, you can probably only resolve about 100 to 200 types. And a similar assay, which instead of using specific reactions for each type, is a probe assay. A DNA probe is mixed with the patient or donor DNA, and if it binds to it, it identifies the variant. But again, well, that's a quick assay. Only about sort of 200 types can be resolved in practice by that method. To identify all variants, you need to sequence. And the next generation sequencing systems are fairly rapid, but they still take a few days to complete. So they're certainly not suitable for deceased donor testing. And there's also a fairly high cost. It's not a cheap method. But again, the resolution isn't perfect. I mean, you can get very high resolution, identify, say all the roughly known 25,000 types, but in practice, it probably is almost a tenth of that. What is being developed and is just about becoming available is a very rapid sequencing method called nanopore sequencing. It's about a 5 or 6 hour turnaround time, but this will resolve all known alleles. So we can identify all different HLA forms. There's a variety of methods. How can we identify the antibodies against them? The things which cause problems in transplantation. Well, as I mentioned, we can form antibodies if we're exposed to non-cell tissue. So here's a sort of a case I've identified, a recipient in green who has a particular tissue type written there, a donor above in red. I've marked the mismatches in with asterisks. So perhaps a transfusion from the red donor into the green recipient might stimulate antibodies against these mismatches, the A2, B8, B44, C16, BR17, etc. How do we identify them all? Can we identify, do we need to sort of test all 25,000 different known variants? Luckily, the answer is no, because of how this variation, this polymorphism works. What I've done here is sort of put down some imaginary types, a red one, a green one, blue one, etc. I've given letters instead of numbers. And this just illustrates how the variation of polymorphism works. Each molecule can vary in about 6 different positions. These are proteins, they're cell surface expressed proteins, and some of the amino acids varies, but usually there's only about 6 clusters. And that's what these different shapes indicate. So the difference between the red and the blue HLA type is just at this position. That amino acid group in the red, the blue variant has this one. So there's only one difference between these two HLA types at that position. But if you look at the green type, all of these positions are different between the blue and the green type. So it's the particular combination of these variants, these epitopes that define an allele. And these epitopes are what are recognized by antibodies. And the epitope variation is an order of magnitude or less than the number of alleles. So we can actually represent the HLA antigenic universe in only a few hundred different molecules. And this makes testing antibodies against these proteins practical. The most common method and most practical method is a bead array, whereby individual beads are colored and they can be identified as individual beads, and each individual type has a different HLA molecule on it. And the assay runs that you mix these microscopic beads with your patient's serum, and if the patient has antibodies against that particular HLA type, they'll bind to the bead, and then a secondary antibody can be used to identify that binding. This is then run through a machine called a Luminex machine, which is essentially a flow cytometer. Oops, sorry, beg your pardon. And the flow cytometer has a laser which interrogates each bead as it goes through. It'll tell whether it's got antibody bound to it, one color, and then the other colors will identify which individual bead, which it'll address the bead. So a positive reaction could be shown and identified of what HLA that bead carries, which is bound by an antibody. And the results look like this. This is an array for HLA class 1, A, B, and C, and shows the reactions. And this is just a histogram which measures the fluorescence of each individual bead. And as you can see, this is actually quite a simple reaction. It's got 3 A2 beads in this array. They're different forms of A2. All 3 are positive, and if you've got good eyesight, you can see that this bead with HLA 69 on it. So there's two critical features about this result. Firstly, we identify what's positive. This patient should not be given a transplant from a donor who's got A2 and 69. But equally important, it's identified the negatives. It's a very, very sensitive assay, this. But the antibody profiles of patients tend to be a bit more complicated than that. And it's really for this reason, because of the epitope sharing, these clusters of variant amino acids. Let's take a situation where you've got a person with a red type having, say, blood from a blue type, so exposed to a non-self blue antigen. The person with the red HLA can then produce antibodies which will react with this epitope, which is the only difference. But of course, the grey HLA type also has that epitope. So you can be immunized with blue, but get antibodies against grey as well. So when you go back to this setting of a transfusion from red into green, you don't just produce antibodies against the mismatches, you produce antibodies against the cross-reactive epitopes as well. So typically what you see in a person who's been sensitized is quite a broad level of sensitization. So this, if you can compare this with the previous result I showed, again, you've got reactions against HLA-A2, but actually this person had a lot of antibodies binding to HLA-B and actually some against HLA-C. The other feature of this, of course, is not just what's positive and what's negative, it's also the level of antibodies. It's a semi-quantitative assay. Transplanting across these levels of antibody on the left, up to 20,000 on this scale, would probably cause immediate hyperacute rejection. But down here, these antibodies are weak and they don't necessarily cause rejection or actually cause long-term problems. On the sort of other end of the sensitization scale, you have a patient like this who's profoundly sensitized. In fact, at the A locus, there's only one, the A3, which is negative, and that's this person's type. And probably you would only find about 1 in 10,000 donors, random donors, would be compatible with this person. And we do see patients like this. So in practice, how do we find out whether the person's compatible or not? Well, the answer is a cross-match, and there's two ways of doing it. There's a cellular cross-match where you mix patient serum with donor cells, mix them together, then detect whether you've got binding. And it's actually very similar to the bead assay, this. Instead of using beads, we're using donor cells, donor leukocytes, which are carry HLA. And we use a flow cytometer to see whether each cell has got a bound antibody from the recipient. And that's a fairly rapid assay. It takes about 2 or 3 hours to do. The alternative is to do what we call a virtual cross-match. If we know the antibody profile of the patient and we know the donor HLA type, we can compare the two. As in this sort of situation, again, this is the very highly sensitized serum I showed a minute ago. That's the recipient type up here, and the potential donor type, and I've just indicated the mismatches in green. And on the Luminex plot, the blues identify the patient's type. The mismatches are in green. As you can see, all the mismatches correspond to the negative position, negative specificities. So this is a negative virtual cross-match. Okay. So there can be two approaches to test compatibility. How do we apply all these tests actually in practice? And what I'm going to go through now is the 5 settings of essentially how we use HLA type to support transplantation, the HLA testing to support transplantation, kidney transplantation. The first is directed donor live donor transplantation where there's a directed donor. And to be honest, the only question we need to ask is, are they HLA antibody compatible? And this might be a family member. And so the simplest method might be just to do a flow cytometry cross-match. If it's negative, proceed. If it's positive, then that is a transplant veto. And it doesn't need any, in one respect, any more testing than that. The virtual cross-match is an alternative, can be done, but this requires the patient's antibodies to be characterized and the donor HLA type known, as I've just shown you. There's actually no absolute necessity to HLA type the patient. There are a couple of issues with this. One is the flow cytometry cross-match is not as sensitive as the virtual cross-match. The detection by Luminex is much more sensitive than by leukocyte flow cytometry. But on the other hand, the very low levels detectable by Luminex aren't necessarily clinically significant. Not in every case. They're not always a contraindication to a transplant. But both methods are of sort of significant practical use. Of course, the other issue is if these are positive and you just have one directed donor for a recipient, how do you then proceed? How do you then get this patient transplanted if their one donor is antibody incompatible? The second setting would be deceased donor transplantation. And how you use HLA depends on your allocation criteria. So you could allocate on the basis of waiting time. The person who waits the longest gets the first allocation or the first chance of when an ABO compatible donor becomes available, or similarly clinical urgency. And if you have a waiting list sorted by that's a priority, then you'd cross-match the person at the top of the list. So again, you don't necessarily need any HLA type. You can use a flow cross-match to see whether they're compatible, or you could do a virtual cross-match if you do have antibody data and from the patient and HLA type from the donor. Another allocation approach could be finding the best match. The better the HLA match, you tend to get less chance of rejection, and overall survival can be enhanced the better the match. It's marginal with the very effective modern day immunosuppression, though. But certainly rejection is more likely with a mismatch. But of course, to do that, you need to have HLA type the patients on your waiting list, and you need to HLA type the donor. And you still need to do a compatibility test, a cross-match by whether it's flow or virtual cross-match. And patients on the waiting list really do need their antibodies tested routinely, so you know what you're going into when a donor is available because of the time restraints of cold ischemia time. And then, of course, the final criteria could just be whether you're interested in compatibility or not with the deceased donor. And that would just need a compatibility test. Could be a flow cross-match or a virtual cross-match. If you're just doing the flow cross-match, then HLA typing is not completely necessary, but in practical terms, you would then be faced with cross-matching a large proportion of the waiting list, of the ABO compatible waiting list. But again, for a virtual cross-match, you would need the antibody data and the donor type. Virtual cross-match can be quicker if cold ischemia times, it's certainly in the UK where there's national allocation, identifying the offered donor as quickly as possible does reduce the cold ischemia time, and a virtual cross-match does help with that. The third application is really combined with the previous one, but when we're testing patients on the waiting list for antibodies. And the issue here is that, well, there's two things. One is that the level of antibody, the sort of broad of the reaction of the patient's serum does indicate the chance of a negative cross-match. So you might have a patient whose antibodies calculated would react with about half your donors. That would be a PRA of 50%. So generally speaking, there's a 50-50 chance with each potential donor. A patient without antibodies, of course, could accept any ABO-compatible donor. Patients, when you're up to sort of 99 and approaching 100%, are obviously very, very difficult to transplant. The other issue is antibodies can change with time, both the specificities and the status. You can go from negative to positive and positive to negative. So when should you test? Well, after any known sensitizing event. Certainly in the UK, we have a protocol that patients are tested every 3 months. This is because the sensitizing events, such as a transfusion, are not always known. And of course, you might want to test at the time of transplant. And if you look on the right of this slide, these just show that I've picked 3 cases which shows how antibodies can behave. On the top is a patient who had was started off as 0, had a transfusion, produced a spike of antibody, and that lasted less than a year. Within about 6 months, it was back down to 0. And it may be safe to transplant across antibodies which were previously detected, but are now consistently negative. Other patients we see have been negative, 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 have a transfusion, they produce antibodies, that might decline. Maybe some more transfusions boost these responses, and that antibody can persist for a little bit longer. But over years, and you can see that this patient took about 5 years for their antibodies to disappear. Other patients almost have antibodies which never go away. In this case, the patient produced antibodies following their graft loss. The antibodies produce a reaction, sort of panel reaction of about 80% against the donors, then slowly started declining, but then had some transfusions around about 4 years later. The antibodies then went up again, and really for the last, actually this was done some time ago, but probably, that patient has had these high level of antibodies for about 10 years. So different patients, due to perhaps how they're sensitized, their antibodies can persist or non-persist in a rather sort of unpredictable way. So that's why, certainly in the UK, we monitor every 3 months. The fourth application is incompatible pairs, and this answers the dilemma potentially for when you have an incompatible pair. If you can find 2 incompatible pairs, but if you exchange between the pairs, in certain circumstances, you can find that you can get compatibility across the pairs. This obviously will require recipient antibody identification. You'll need a donor type, and then a cross-match, either a virtual cross-match or a flow cytometry cross-match. But paired kidney exchange is proving a very, very successful approach. And the third application, sorry, I beg your pardon, the fifth application is post-transplant antibody testing. Following a transplant, rejection is not uncommon, depending on the type of patient. Usually for unsensitized patients in the UK, we see it in about 5 to 10% of cases. Rejection, of course, can be diagnosed histologically, but part of that diagnosis would involve finding out whether there are donor-specific antibodies or not. And hence you would test to see whether the patient did have antibodies against the current donor. And of course, it can guide the treatment. If the rejection is associated with antibodies, you may want to remove the antibodies. If there's no donor-specific antibodies, then that form of treatment of the rejection would not be necessary. So this requires obviously recipient antibody identification. We need a donor type. It's quite useful to have the recipient HLA type, as the Luminex test is not foolproof, and we sometimes see false positive reactions, and the recipient HLA type can help resolve those sort of problems. So in summary, I've got two summaries. One is summarizing those last 5 slides, just to show that we have a variety of tests. We have HLA typing, identification, and cross-matching, how we apply those to the different settings. And red means, so the red-pink color identifies where you need to do the test, green is optional, and blue depends on the context. So you can see, obviously, for deceased donor allocation and paired exchange, the full repertoire of testing needs to be applied to those settings. Directed live donation, the minimum you would require is just a form of cross-matching. Obviously, if you're doing a virtual cross-match, you need a donor type and the patient identification of their antibodies. HLA typing is not absolutely necessary, but it does help resolve problems in identifying antibody specificity sometimes. So the point about this is that depending on the context, depending on what methods you may want to use. And finally is a skills matrix. People. We've got methods, but we need people. And for flow cross-matching, obviously we need flow cytometry skills. For a virtual cross-match, we need antibody detection, which is essentially Luminex, and molecular biology skills. Antibody identification, the Luminex platform, you need those skills. There are other ways of measuring antibodies, but Luminex essentially is the sort of the universally accepted method. For HLA typing, we need molecular biology skills. Certainly that's PCR polymerase chain reaction. Actually, and the Luminex machine can be used for HLA typing as well. The knowledge and expertise we need for this, we need technical expertise for all these areas. These are all laboratory testing procedures. We need scientific knowledge to be able to understand virtual cross-match identification of antibodies and assignment of HLA types. And we need clinical expertise to interpret cross-matches, and whether it's a virtual cross-match or a flow cross-match, in order to use this information and transplant safely. So I hope I've kept reasonably to time. That's my presentation. So thank you very much for inviting me. Thank you for NHSBT, who pay my salary, which is good of them, and for their support. And particularly to the Commonwealth Tribute to Life and Mohun Foundation for organising this meeting and inviting me. So thank you.
Thank you, Dr. Briggs, and I think it was a beautiful presentation. I'm sure we learned from that. Are you ready to take the question at this moment?
Yes, definitely, yes. Delighted to, thank you.
The first question, and I'm very much interested in this question because of our setting in the Caribbean. And it said, what advice you have for someone setting up a laboratory in a country without any of the any such tissue typing laboratories?
Yeah.
And I see it was written that Dr. Sunil Shroff willing to answer that question.
No, I'm sorry, Kishore. It will go with the speakers are going to answer. But my request is, if you don't mind, we'll take Dolly's presentation and do a panel discussion after we finish Dolly's presentation. But there may be some overlaps. Is that okay with Dr. Hasina and you?
Yeah, that's okay.
Yeah. I mean, I know there are very important questions, and we may go a little bit over time today, because I know this topic is a little complex, and we will need a little bit more time. So after Dolly has done the presentation, I know we'll open the floor and then have a good panel discussion.
Thank you.
Thank you, Kishore. Thank you, Dr. Hasina. Dolly, you're on.
Yeah, so, yeah, sure. So are my
Hi.
slides okay? Is it visible?
Looks good, Dolly.
Yeah, okay. I think I should thank Professor Briggs because he set a good platform for me to go about describing our journey of setting up a transplant immunology lab in a country like India. I think a talk of this would really be inappropriate unless it's put into context, because India is an extremely diverse country with a lot of diverse problems as well, whether it's its massive population or its number of people below the poverty line, the massive challenge of inequity, and the huge rich divide poor. And against this, when we are still battling things like maternal mortality, infant mortality, uh, we have to prioritize what we want to do. And you can imagine that in the hierarchy of priorities, setting up a transplant immunology lab would probably fall very, very low. However, I think it is very important to look at any kind of a problem with varying perspectives. And I think the important message to us was that transplantation in our country was here to stay. It had become far more affordable than many decades ago. So the question is, how can we make it safer? And how can we make it most affordable for the common man? Because in the long run, if there is greater graft longevity, it ends up in financial savings for the patient as well. Now, when we think of setting up a laboratory, of course, there is a cost of infrastructure, a cost of equipment, the personnel and the training required, and there is a certain cost that gets transferred to the patient in terms of tests that need to be done. And the basic question that we've asked ourselves is there an incremental gain for the patient by doing those tests? And is the lab sustainable with the kind of cost that it costs the patient? I think power to set up these come from need and not desire. And very often it's important to identify that need. For us, the need came in the late 1980s when one of our bone marrow transplant patients, who was supposed to be an identical HLA match, just rejected his graft and subsequently died of acute GVHD within the first few weeks after his transplant. Our hematologist sent the samples across overseas, and he got the question back as to, do you have Portuguese antigens in your population? And then they subsequently had typed it and found that the patient and donor were actually not a match. A hematologist refused to proceed with transplant till we had an in-house laboratory. Now, unlike in UK, we are a very fragmented healthcare system and most of our services decentralized. So therefore, within an institution, it becomes quite challenging to set up certain services. But the question came about as to who is going to be the safest donor, therefore, for any form of transplant, and how do we ensure the best chance of graft longevity? As Professor Briggs showed, I think one thing we definitely want to avoid is this, a hyperacute rejection of a transplanted organ. So we started, and in 1990, we had Dr. Paul Terasaki actually come and start our lab. He came and he conducted a very small workshop, and he initiated us into the world of complement-dependent cytotoxicity. We all know the clinical impact of this test, and though it is subjective, though it is prone to interference with non-HLA antibodies, this was the platform with which our transplant immunology lab started. However, there was a reason for it. We found that we could actually share a lot of facilities within the institution. So within our own lab, other labs, we had centrifuges, water baths, pH meters, storage facilities, and all those could be shared. And we invested only in absolute necessities. So that included a phase contrast microscope, microcentrifuges, a laminar airflow, syringes, Hamilton syringes, and the like. But right to the beginning, the emphasis on quality was immense. Standard operating procedures were put into place. We had everything, including a tachometer, to make sure that our centrifugal speeds were correct. Training of personnel had to be done, and we were enrolled into the UCLA EQA from day one. So though we were doing very basic tests, and from Professor Briggs' timeline, I think we were about a decade behind very, very easily. We also invested in many cost reduction strategies. For example, personnel. So I was a junior doctor after my MBBS there at that time. I was a clerk, I was the storekeeper, and I was a technician, all rolled into one along with one more technical person. We got antisera in vials and actually did the dotting, 1 microliter per well dotting in the lab, because it was cheaper to buy it that way than to buy pre-dotted trays. So many things like this were done initially because we needed to show that this was something which was sustainable, and till numbers picked up, it was very, very critical that we conserved on as much cost as possible. On the same platform, we went ahead and tried to separate B cells so that we could do a split T and a B cell cross match, and also isolate B cell for a class 2 HLA typing on serology. So on the serological platform, for almost 8-9 years after the lab was set up, we were doing CDC cross matches as well as class 1 and class 2 typing. But however, we realized that yes, there were lots of problems with serological typing, and that we needed to move on to molecular. We knew that our cross matches lacked sensitivity. We worked very hard to be able to differentiate IgG from IgM antibodies and to do enhancements on this platform itself. So we did the anti-human globulin cross match, the triple wash, the extended incubation, all with the hope that we would add to sensitivity on the platform that we had on hand. It was a long waiting period, but the important lesson it taught us was that we cannot wait for things to become perfect till we start, and that beginning is extremely crucial. But soon we realized that while we were thinking of cost effectiveness in a monetary way, we needed to think of it in a much more broader sense. I think quality of life years gained is very, very important when we talk about what we offer our patients, and not just about graft survival for the first year or the second year. This is a recent article, a 2020 article, and this is some Irish data. And it talks about patients who come for their first, second, and third transplants. And they talk about how if you have a live donor transplant, they do better. And what struck me was that when our patients come for a transplant in resource-constrained countries such as ours, they come for the first transplant having sold their homes, having sold their property. And most of the time, they cannot afford to come back for a second or a third transplant. So therefore, it was our duty to make sure that the best was given to them right at the beginning. So we realized that while we had embraced diversity and made sure grafts were safe to a point, graft longevity required much more testing. And it was important also that it was with the minimal immunosuppression possible, because in our country, infection is a huge problem. So you can't immunosuppress beyond a point. Patients will die of infection rather than of graft failure. So it was a fine balance we had to try and maintain. As shown by Professor Briggs, the polymorphism of the HLA locus just kept increasing. But we also knew that with the structure of the HLA molecule, there were a lot of permutations and combinations which allowed differences to be acceptable. And for this, yes, we knew we would need sequencing, but we thought it was time we moved into molecular testing. We're all aware of low and high resolution. And while we couldn't get straight into it, we thought we should move down the molecular path. There was enough data to show that better the HLA match, better the outcome. Lack of antibodies better the outcome. So one would think, why not? It's so straightforward, just go to it. But the problem was the multitude of platforms which are available to us. And if you just took molecular typing, you had a PCR-based SSP, you had the SSOP based on the Luminix as discussed earlier, the Sanger sequencing, and the next generation sequencing. So with this kind of a wide variety in front of us, we needed to know what was going to be practical for us. And in this setting, I just want to emphasize that in countries such as ours, we have to think through very carefully about what is the infrastructure that is required, what are the personnel required, does the test give us basically what we want, at least at that starting point, the cost, of course, but equally so, what is the service support, downtimes, and the backup of equipment and infrastructure that is required for these various platforms of testing, and whether there's a presence regionally. So if we have a company set off somewhere in the UK or the US with no presence in India and just importing their machines, every time we have a problem, it's going to be a huge issue trying to sort it out. So the regional presence of a vendor is equally important. And of course, the different financial models of investment, which we'll talk about. So when we thought about a molecular lab, yes, the infrastructure was needed, but equally so between a sequence PCR SSP and an SSOP, we decided first to go with the sequence-specific primers simply because we knew that we could do it without too much investment initially, because there were different areas in the lab, which in the institution, where we could borrow time, lab time and lab space. So we started with the SSP and demonstrated that it was working. And as we worked, we also got experience. We simultaneously thought of setting up a molecular lab. And in this regard, it is very important that when we plan infrastructure, we plan it with reference to professional guidelines. For example, the workflow of a molecular lab should be set in place when we start. So a unilateral workflow with making sure that there's no backflow from a post-amp area, all those are things that we should. It's important also to look at accreditation guidelines for local needs, because finally it is important to get local accreditation as well, and it's important to put this together when we plan our infrastructure. It's also important to make sure that all our assay and equipment validation is in place and our checklists are done. Training of personnel, extremely important. So this is just an example of the unidirectional workflow which is expected in any molecular lab. Now, the other thing we learned the hard way was that when we decide on a certain assay, we need to check as to what equipment that assay was validated on. A simple thing like a Luminix-based assay was not consistently workable on an Eppendorf thermal cycler. It required an ABI one. And therefore, it is important that when we plan, we do our basic homework, when we choose our thermal cyclers, electrophoresis systems, and such. I think a backup is something that we need to keep in mind all the time. It's important to pay attention to the nitty-gritty, what kind of gloves we need, what kind of filter tips we need, what size filters we need, and also everything, including our electrical connections, need to be 100% right to make sure we don't have a short circuit somewhere. So the evolution of our HLA typing over a period of about 25 years went from serology, which stayed with us for almost 9 to 10 years, to molecular, which was the SSP and the SSOP. And finally, right now, we sit with an NGS-based typing, which has just been in use for the past 3 years. Our numbers have slowly climbed over these years, and now we do batches of 32 to 48 samples every week. And it has proved to be a very sustainable initiative. Now, side by side, we knew that antibody testing platforms needed to be enhanced as well. And again, the same problem, so many available out there, and it needed, and with varying degrees of sensitivity and varying degrees of clinical impact. And we needed to choose the most clinically applicable ones. In this regard, I just want to highlight that there are different formats as well as different platforms. So we have an antibody screen, cross-match, a virtual cross-match, and a panel reactive antibody. And we have these platforms, starting with the CDC, which is the least sensitive, to the ELISA, flow cytometric, and Luminex, with the Luminex being the most sensitive. So for us, for example, an antibody screen was something we had to choose, and we had to prioritize, and we had to define cost effectiveness. So an antibody screen was definitely out because it just told us if there was an antibody or not. What we really wanted to know was whether there was a donor specific antibody. So it was a cross match, whether physical or virtual. We also decided to go with the Luminex to start off with, because the Luminex had advantage of being able to do a HLA typing, and with increasing numbers, it was an easier platform to navigate. And apart from that, it could also do antibody testing. As Dr. Briggs pointed out, this is the virtual cross-match with single antigens attached to individual beads which had unique fluorescent signatures. However, when we started off, it was exorbitantly expensive. And only a few years ago, we came across the half reaction protocol by which Dr. Claus's lab had validated. And that has cut cost and made it far more accessible. So while we were dealing and juggling around cost issues, this is just to show you what it costs. A complement-dependent cytotoxicity is 30, and it goes up to a single bead, which costs us in India almost $500 if you do it in its full reaction. Just about the same time, the concept of a Luminex cross-match, where we use donor material, donor lysate material, came. And there was a certain applicability to transplant. So we started to use the Luminex cross-match, but it had lots of gaps. It did not pick up DQ antibodies, it did not pick up C antibodies, and therefore you could miss it. But it was a gap in the middle. The moment we got a flow for some other reason within the department, we standardized the flow cytometric cross-match because the clinical applicability to it is great. It's important to also understand that each platform comes with its share of trouble. For example, if a patient is on rituximab, you have interference on the flow, but pronase can clear it for you. We found that flow in our setting is very prone to the prozone phenomenon. So we had to be careful, but because we always did a virtual cross-match in tandem, it helped us pick up these. This combination of tests actually helped us a great deal because it helped us stratify our patients in terms of immunological risk, and this was particularly useful in a deceased donor setting. Patients coming for a second transplant, it always insists on the typing of the first donor, and it's very important in terms of choosing the second donor. We realized that yes, we should follow guidelines, but very often we should allow flexibility within our system. And therefore, looking at the Transplantation Society guidelines, we've modified it the Indian way to allow it to be feasible in our system. So now what we do for our renal transplants is a high resolution NGS-based HLA type, a good enhanced CDC combined with a flow cytometric cross-match and a virtual cross-match. If somebody is on the diseased organ donor list, then they have a virtual and a single antigen bead assay done every 3 months or every 6 months. The single antigen bead assay is very, very talked about and is sort of considered a gold standard. And we too have been very excited by it. But we found that for the Indian population, it is a challenge because more than 50% of our alleles are not actually represented on the panels that we get. And this is a recent publication from our center in Transplant Immunology where we've discussed this. We really hope that an Asian panel, which is more suitable for our populations, will come in. But with these extremely wide array of highly sensitive tests picking up antibodies, it is very important that we don't end up going on a wild goose chase and not knowing what to do with what we found. I love Dr. Claus's statement who says that antibody testing should be to facilitate and not to prevent safe transplantation. And there's this very elegant study in the NEJM some few years ago to say that even in those who are highly sensitized, the outcome is better when transplanted against an antibody than those on the wait list. It was a huge learning curve with deceased donor transplants, and I'm really grateful to Dr. Shroff, Dr. Georgie, and Dr. Amalur Pavnathan, who actually introduced me to this whole concept and initiated us working with organizations outside our institution, created huge clinical lab interfaces and networking and attempting to harmonize practice across the state and even outside has been a really, really huge learning experience. And I think truly they've not just thought outside the box, but almost got rid of the box. So when we get a call for a deceased donor kidney, this is what we have. The donor is already HLA typed on the NGS platform. We have a Luminex-based typing done for the donor then. The CDC and the flow is done at that time. And if we have a single antigen bead assay done within 3 months with no sensitizing events in the interim, then that is used to decide suitability of the donor. This combo sometimes seems like a dream come true, especially when we think of our days of CDC. But I think nothing remains constant except change. And we deal with unexplained rejections and are dealing with non-HLA targets at the moment on a research mode to see whether any of them have greater clinical applicability in our setting. We're also looking at epitopes to see whether some mismatches are more forgiving than others, the role of complement. The few lessons we've learned along the way. The first thing is that we have to be ready for change. And it is very important to make that change earlier rather than later when it's feasible. Moving out of our comfort zone is absolutely critical. I trained at the Royal London for 3 months before I initiated molecular typing back in our institution. And I know how petrified I was of it. And the number of times Dr. Sinat must have got calls from me subsequent to us initiating it. Also, when we are prioritizing and choosing tests, we need to be very careful. Are we plugging the right hole? We want a sensitive antibody testing platform, choose a cross-match rather than a screen, those kind of things. But in that process, we also thought that we should try and initiate strategies which will prevent antibodies from forming. And leukodepletion is something that we've advocated very strongly for patients who are likely to go in for transplantation. Keep the clinicians on board with you and your lab will grow. Because finally, it is the response to a clinical need. Many people have asked me, who's the clinician, who's the lab? I think the important thing is that both seem to be living in happy harmony here. And this is our clinical team. I don't think anyone looked very crocodilish. It's important that we learn to function with limited investments, prove our sustainability before we can actually take things further. But decide on our non-negotiables, one of the most important things being quality. Models of financial investment should be thought through. Very often, we don't actually do an outright purchase. We ask for reagent rentals or trials on a research mode, and then we get into more definitive systems. A hub and spoke model is a good thing to follow, particularly when our numbers are small. And I think in this whole process, we will definitely be able to offer high-tech, appropriate care in a low-cost, effective setting. This has been our journey from 1990 to now, clearly quite behind in many decades as we trudged on. But I think this journey has shown us that in some small way, we have reached the point where there is a certain degree of equity to our patients who are going in for transplantation in an affordable manner. And I think this is very important because accessibility to safe patient care is truly an issue in justice. I don't think this is possible without the number of people before me, and I'm sure who will be there after me, to take things forward. And I'd like to just end with a very favorite quote of mine. It's from a footballer who told his team, which was, in a losing spree, saying perfection is not available. But I think if we relentlessly chase it, we will definitely catch excellence. And I like to think that we are relentlessly chasing it. So thank you.
Thank you. Thank you, Dolly. We are just a little bit over time. And I think, we'll extend the webinar by 15 minutes, but there are many questions in the chat, question answers. So I hand over to Dr. Hasina and Dr. Kishore to take the questions with the speakers. Thank you.
Thank you very much. Are you all hearing my audio?
You are audible, yes.
Hasina, go ahead.
Great. Thank you. So I will start. So thank you very much, Dr. Daniel. And that presentation was very excellent. I think it's a great lead up to our first question, which is what advice would you have for someone setting up a laboratory to facilitate transplant in a country without this option at the moment? And this is open to all speakers and experts.
If I make a start, I think Professor Daniel has actually probably answered it pretty well. Yes. I think you start small, you build up until, as you said, you reach that excellence. The trouble is very easy for me. In the UK, we've had an established program. Our ways of working have developed over the last 40 years, and we have a national program. I think our testing is probably excessive. But I think if you aim at what questions you need to answer, do you have a donor recipient pair? Are they compatible? What tests do you need to answer that question? And it may just be a flow cross match. And you may have those facilities and skills already existing. As you get more complicated in terms of allocation, then you need to add to the testing. You need to put in HLA typing and antibody testing. It becomes requiring more skills, but also more expensive. And I think you need to build up those skills and knowledge. But unfortunately, I came to a system which already existed, so I find it very difficult to know how it actually works, starting from scratch. I don't know if Professor Daniel has a better idea than I do on that.
Dolly?
I think the important thing is that I agree with Professor Briggs that I think the critical question is what is the most important test at that point? And I think if you combine it with some facility which is available within your institution, then it makes it easier to implement. It may be very tiny steps, but it's okay.
Yeah.
So I don't know whether it answers your question.
There's no perfect answer to that, but yes, you have to make a start. And it also depends on your volumes as you go forward, whether you can sustain it. That's, I suppose. You want to take the next question, Kishore? There are many in the chat window. You need to finish these questions.
Dr. Hasina will be asking the question because I'm unable to see them at this moment.
I will ask the question.
Yeah.
Are some HLA antibodies more of a problem than others? And or are they all just as troublesome? Prof Briggs, do you get the question?
Okay, Professor, yes, a very, very good question. I think the answer is there are differences, but we actually don't quite fully understand. And there's two issues. Some antibodies, HLA-C has lower expression than A and B, so you can probably the patient could tolerate higher antibodies against C than they could A and B. High levels, that's measured by a cross-match or certainly by Luminex. The other issue is what stimulated those antibodies. If a single transfusion stimulates antibodies, they tend to be fairly weak and short-lived. Transplanting across those is probably safer than if you were transplanting across a similar level of antibody, but if it was stimulated by a failed graft, that usually has more significant consequences because of the underlying immunology. Pregnancy is a bit odd, only affects only a proportion of the patients, of course, but it does appear that you can transplant reasonably safely against previously simulated, pregnancy simulated antibodies, but once they've disappeared. So there's a time issue. There's actually the issue of how they're stimulated and also their level. I hope that helps a bit.
Yeah, thank you.
Thank you very much. So the next question is, are bead arrays expensive, and what would be the cheapest or lowest resource way of detecting HLA antibodies?
Who will take that? Dolly, will you take that?
I feel if we are doing a single antigen bead assay, for example, there is a PRA calculation which comes out of that. However, to get what is called a calculated PRA, which is relevant to our population, becomes quite complicated because we don't have local frequencies. Now, a PRA, I think, assumes maximum significance when you have somebody who's sensitized and on a list waiting for an appropriate donor. Otherwise, it just defines a breadth of sensitization. And it tells you, like in somebody who is not highly sensitized, I don't think it adds too much value. I think it is more critical to go in for a cross match. Would you agree, Professor Briggs?
Yeah, no, I think you're exactly right. Yeah, I certainly do. Yeah. That's a good point.
You sure? Your next question you want to take? I put it in the chat window.
Sorry, I'm unable to see any of the questions.
Okay, I will help out here. Hasina, can you see the questions?
I can't see the questions, Dr. Shroff. So the next question is, do you ever get caught out by virtual cross match and find cross reactivity that you didn't expect?
Yes.
Can happen.
I don't know if you find the same, Professor Daniel. And typically, it's where we don't have the high enough resolution of the HLA type. And those are the cases where we say we cannot identify without any doubt that those antibodies will react with that HLA type. So it's the resolution of the typing sometimes lets us down. We usually can see it up front, so we don't get caught out and make a mistake of transplant unsafely. But in those circumstances, we'd probably do a flow cross match, a wet cross match. Does that?
Yeah.
Thank you very much, Professor Briggs, for answering. So we'll move on again to our next question. So the next question is HLA typing like blood groups, where you need an up-to-date sample in order to make a safe cross-match? And do patients on the waiting list have regular HLA typing done, or is it less critical than that? That's two questions there. Is it similar to group typing? And should it really be done regularly or just when they're having their transplant?
Did you get that question?
Yep.
Does anybody want to take it? Maybe Dr. Daniel?
So is the question whether they just do a HLA typing like an ABO blood group? Is that the question?
So they're asking if you must have an up-to-date sample. So it's similar to when you typing blood groups as for a group and cross match. Do you need to have a new sample each time, or is it that you can use an old sample from before?
No, we just type once, and it is there for reference subsequently. We do not repeat HLA typings. That's our policy. I don't think we can afford to either.
Yeah, I agree. It doesn't change. We tend to type everybody twice as a confirmatory sample, just to avoid sample mix-ups. But once we've assigned the type, we assume it hasn't changed. It's accounting.
There is one question to Professor Briggs. There's Luke Foster, I think he knows you, and he says, "Thank you, David. Do we have any idea why some people may modulate their antibody levels, whereas some may persist?"
I think Luke knows very well. I don't know the answer to that question. I mean, it's a really good question. It appears, I mean, I think a failed graft tends to result in persisting antibodies. I guess because you've been exposed to the mismatches for so long, that it generates a really sort of deep immune response. But no, Luke, we don't always know. We're trying to investigate it. I don't know if Professor Daniel has any insight into it, but the answer to the question, when we have patients who've been on the waiting list for years, sometimes decades, without their antibodies changing, we'd really like to know how to sort of manage patients like that. And whether 20 years ago, we could have said to them, no, you won't lose your antibodies. We need to think of a different strategy. But it's one of the big questions we need to answer.
Thank you very much. And Dr. Daniel, any input into that question? Or did Professor Briggs cover it very well?
Yeah, absolutely. Yeah.
Excellent, great. So our final question for today is, how important is minimizing HLA mismatches to reduce risk of sensitization in cases where retransplant may be required?
Shall I start on that, Professor Daniel? And
Yeah,
I'm interested in your comments. Yes, I mean, it'd be great to do that. To be able to do that, you need to have access to a large enough donor pool to be able to match well. And obviously for directed donation from a family member, you don't always have that. They may be well matched, they may not be. So it's almost the luxury of having a wide donor pool allows you to do that. Because but also need a setting where retransplantation is an option. And Professor Daniel, I think you referred to that. Economically, it's not always possible in some settings that actually a second transplant will be possible. So maximizing the success of the first transplant is so important, and I know you mentioned that, Professor Daniel. But if you can do it, yes, but you need the luxury of having a wide enough donor pool.
We don't even manage to get a HLA match. If it's a family donor, sometimes a lot of the time it's a haplo match. But anything more than that is almost never seen. And we focus on getting the antibodies right and making sure that there are no antibodies, I think.
There's one question I think which we may have missed. I would perhaps take that and we can finish after that. And this question is about, are bead array expensive? And what would be the cheapest or the lowest resource way of detecting HLA antibodies? Good question. Dolly, you can answer that perhaps. What is the cheapest for? Are they expensive?
I don't think anything in transplant immunology comes cheap, Dr. Chaw, unfortunately. So, but I think if you look at it as a trade-off, for us, a half reaction protocol of the single antigen bead has worked very well. We have done a large series of comparisons after looking at Dr. Claus's paper, and we haven't missed any. So we now use a half reaction protocol, which has brought it down. But having said that, a virtual cross match alone does not suffice in our Indian population. We need a physical cross match, and therefore a combination of a flow and a single antigen bead. And in that, it works out to about 20 to 25,000 rupees, which is about 250 pounds.
Yes, so, or $300 perhaps.
Yeah, so that's the kind of cost that it costs for a good antibody screen.
So shall we get Kishore to give some closing remarks since he has not had access to the questions? Kishore, over to you.
Thank you, everyone. I really appreciate this gathering, and I'm happy to meet all of you here, and it's a pleasure because I have one question to everyone, and this is the reality of facing the transplant program among the Caribbean country. And I'm sure Hasina and all the other members from the Caribbean here will agree with me. One of the challenges that we have in developing our program and advancing transplant in the Caribbean is to get our HLA typing done and our flow cross match done. And that's because there's no country in the Caribbean other than Trinidad, where Hasina is from, that was doing the cross match. No other country is doing actually flow cross match. Most at present, we have to send our cross match to the US, and we have to FedEx it. So my question here: what are your advice, and how much would it actually cost one of the Caribbean country here to set up an immunology lab, and how do we move forward? I'm I'm desperate for that, and somewhere is that who can help us to develop, because we cannot move forward. All we do is live in donor transplant. We in Guyana and Trinidad has the legislation, and there are more Caribbean countries who's pushing for have the legislation to advance cadaveric transplant, but we are still late at this moment in moving forward.
I think it's very pertinent, and I think not only you, many African countries also in the same kind of situation. I think, we can get Professor Briggs and then Dolly to really give some comments on this. I know it's not an easy answer, but we don't have much time, maybe half a minute each, perhaps.
Yeah, I agree with the problem. I mean, the geography doesn't help the Caribbean because you're islands, and moving samples around is a problem. My feeling very, very quickly, to start with, using a cross-match technique like flow cytometry will allow you to do live donor transplants safely. If it's a directed transplant, taking it on to deceased donor transplants, you do need a local lab because you have to do HLA typing. But directed live donation actually requires basic sort of flow cytometry skills or for the cross-match. But otherwise, it is expensive. Yes, Professor Daniel.
You've been through the whole cycle, I know.
So we've actually put together some kind of a costing, Kishore, for setting up of basic transplant immunology services. I'm not sure how it translates in the Caribbean in terms of costing, but I'm happy to share that with you in terms of our running and infrastructure costs. And maybe we can discuss this offline to see if there's some way we can actually take it forward, because we have helped local countries, nearby countries, sort of handheld a little bit to a certain extent. So happy to work with you if you'd like.
I think
thank you.
Sorry to interrupt, but I think the good practices document this will be very important for the Tribute to Life project. And Dolly will be looking forward to something like this, which we can put it on open source, so that some of these countries, we have 54 Commonwealth countries, of which about 25 or 30 I think would want to go on the transplant area, and of all these 25 countries, there are only very few handful which are actually having a immunology lab. So I think it will be a very useful exercise, and Kishore's question is very, very pertinent. It will apply to many, many, many more countries in the Commonwealth. So Hasina, do you want to say anything before we close?
Sure. I would like to thank the Commonwealth Tribute to Life for the opportunity and for hosting in collaboration with the Mohan Foundation. It was an excellent topic, and like Dr. Prasad said, in the Caribbean, we are trying our best to set up our programs. They're all very young still, and they need a bit of guidance and a big brother. And I think this is the best opportunity for it to happen. I just wanted to thank the speakers, Dr. Daniel and Professor Briggs. They were excellent as well. And thank you very much, Dr. Shroff, as well, for having us.
Thank you, Dr. Hasina, and thank you, Professor Briggs. Thank you, Dolly, for stepping in and doing such an excellent job on setting up a lab, especially for developing country. Dr. Kishore from Guyana, Dr. Hasina, I think, and Laurie, of course. Laurie has had to step out. He had gone to his intensive care. So thank you everyone for being here. I know we have overstepped a little bit, but this is such a complex topic. I think it required an extra time to do justice to it. and both the speakers did an excellent job. I mean, remarkable. Even I in fact, I'm a transplant clinician surgeon. I learned a few things myself, especially the Professor Briggs. I don't know when that is going to happen and what kind of costing is going to be for that kind of thing to happen. But I think when that happens, I think there'll be perhaps more advancement towards, perhaps we'll see less rejection as we go forward and better long-term graft survival. I don't know. I'm still, I mean, it's still new to me. So hopefully we'll see that happening, perhaps over the next few years. So with that, I will close this webinar. Next week, again, same time, Thursday, we will have, discussion on opt-out legislation, especially with England going in towards the soft opt-out legislation. I think is a very important topic. So same time next week, I think we started at 4:30 today. So same time, India time, and same time, I think UK time will be about 12. Am I right, Professor Briggs?
I think so, yeah.
Next week? About noon, perhaps, next week, or is it 1 o'clock next week? Same time.
Check.
4.5 hours, yeah. So it will be noon time next week for England, and I don't know about Guyana and Trinidad, Hasina, and Dr. Kishore, but as international panels, kindly keep your local kind of hospitals and local people informed if they want to attend this webinar. And also remember, these webinar series are available on the Tribute to Life platform for everyone to watch. So with that, I'll close. Again, thank you very much for attending, and I hope we answered all the questions. Thank you.
Thank you.
Thank you
Thank you very
much.
much.
Bye-bye.